transplantation antigens. The transplant tissues which are not rejected or which do not induce an immune response are called histocompatible. Histocompatibility results due to antigenic differences. The antigens which play a major role in transplantations are encoded by more than 40 different loci. They are broadly classified in three categories as blood group antigen. The ABO blood group antigens represent major blood transplantation antigens. The ABO antigens are polymorphic in nature and are present on the surface of erythrocytes. The blood group alleles A and B code for different sugars on the RBC surface. Blood group O is null allele and does not code for any sugar, whereas AB represents both alleles. These alleles are codominantly expressed and follow Mendelian inheritance. An individual can either be heterozygous or homozygous for the inherited alleles. The reason why these are called as transplantation antigens, we all have isohemagglutinins, like antibodies to these blood group antigens. These antibodies develop as a result of cross-reactivity to certain ubiquitous microbes. A person having blood allele A possesses antibodies for B allele in its serum, and if such a person is transfused with blood carrying B allele, hemolytic reaction mediated by type 2 hypersensitivity reaction take place. Transplantation rejection. Isohemagglutinin A anti-B, B contain anti-A, AB contain none, O contain anti-A and anti-B. Remember like that. Kind of similar to antibody. So major histocompatibility complex antigens. For the transplantation of nucleated cells, the major histocompatibility complex antigens are the major barriers. The MHC molecules are present on all the nucleated cells, and the primary role is to direct T cells to carry out their function. Just like the blood group antigen coding locus, genes coding for MHC molecules are also polymorphic. There are 6 internal loci and a very large number of different allelic forms exist for MHC. The MHC complex in humans is called a human leukocyte antigens HLA complex and is present on chromosome 6. In mice it is called a H2 complex and it is present on chromosome 70. The MHC loci are closely linked and thus the individuals inherit the alleles encoded by these closely linked loci as 2 sets, one from each parent, where each set of alleles is referred to as haplotype H2 complex, HLA complex. The varied combinations of many different allelic forms which are codominantly expressed ensure very rare chances of 2 individuals having identical set of alleles and thus form the basis of alloantigen rejection. Major histocompatibility complex antigens. Beside the MHC, there are many minor histocompatibility antigens also which are usually weaker and usually targeted in late onset rejection. The tissue rejection, major histocompatibility complex antigens. The tissue rejection due to minor histocompatibility difference is less vigorous than major compatibility differences, but still they may lead to graft rejection, and that's the reason why even successful transplantation between HLA identical individuals requires some immune suppression. The non-ABO blood antigens and the HY system thus are present in Y chromosomes and hence are expressed only in males. Antigens are some of the best studied minor histocompatibility antigens. HY system, ABO blood group. And allograft rejection mechanisms, hyperacute, acute and chronic. Depending upon the organ or tissue being transplanted and the immune reactions involved, graft rejection have different time courses and accordingly they are classified as hyperacute rejections occurs within 24 hours of transplantation. Acute rejection begin in first few weeks after transplantation, then chronic rejection start from months to years after transplantation. Occurs within 24 hours of transplantation. Hyperacute rejection begin in first few weeks after transplantation. Acute rejection, then chronic rejection. Chronic in the sense start from months to years after transplantation. So that is chronic. Hyperacute, acute, chronic. Chronic is kind of relaxed. Okay. Then hyperacute rejections. If the host serum has pre-existing antibodies against graft antigen, the transplant is quickly rejected even before vascularization. Complement activation leading to neutrophil infiltration causes blood clotting in the capillaries that doesn't allow vascularization. Xenoplants are usually rejected in hyperacute manner. The mechanism for presence of pre-existing antibodies for allogenic MHC antigens can be women having repeated pregnancies are exposed to maternal alloantigens on fetus and thus develop antibodies to these antigens. If some of these antigens are same as those on subsequent graft, then a hyperacute rejection is mounted. Then repeated blood transfusion may also cause development of antibodies against MHC antigens on WBC of transfused blood. And if some of these antigens are same as those on subsequent graft, it leads to hyperacute rejection. Then individuals having a previous graft may have antibodies against allogenic MHC antigen present on that graft. In some cases, instead of pre-existing antibodies, individuals may develop antibodies to transplant immediately and lead to less frequent form of rejection called the accelerated rejection. Grafted kidney, pre-existing host antibodies are carried to the graft which bind to antigens present on renal capillaries and reactive complement system. Active complement system. Then split products of complement attract neutrophils which release lytic enzymes. The lytic enzymes destroy the endothelial cells or on renal capillaries. The injured tissue is occupied by platelets which further cause vascular blockage. This is the hyperacute rejection in kidney transplant. Acute rejection. The acute rejection of transplant is manifested by cell-mediated immune system. T helper cell activation and proliferation leads to activation of various tissue effector cells including NK cells and macrophages, which further lead to cytotoxicity. Then chronic rejections. Chronic rejection take place after months or years of transplant and include both humoral and cell-mediated immune response. Tissue typing and immune suppressive drugs have greatly enhanced and short-term survival of transplant, but chronic rejection is not yet understood or managed. Memory response, cellular immunity to HLA and minor transplantation antigens are also key factors involved in chronic rejection. Graft versus host and host versus graft rejection. Most of the transplants are rejected due to recognition and a response of recipient's immune system to donor alloantigens, and this is called host versus graft rejection. But in some cases, bone marrow transplant and additional problem arises where the viable active T lymphocytes in donor tissue recognizes the mismatched HLA alleles of recipient and respond to them. This response is called the graft versus host response. Host versus graft reaction responds to donor HLA by recipient immune system. Then graft versus host reaction responds to recipient HLA by donor T cells. Prevention of graft rejection. Familial grafting, major histocompatibility complex or HLA in humans is the major barrier for transplant acceptance, but due to inheritance pattern of HLA genes, the transplantation within the family members reduce the allele mismatches significantly. Generally, there is very little crossover within the locus and the whole locus is inherited. Thus, there is 50% match of HLA alleles if transplant is from parents to siblings, whereas it's 25% match from sibling to sibling. Hence, familial grafting ensures better graft acceptance. Then cross-matching can be conducted. Cross-matching is done before transplantation to check that there are no preformed antibodies in recipient serum for donor HLA. In this method, serum from potential recipient is mixed with the lymphocytes of donor in the presence of complement for evaluating lysis or stained with fluorescent antibodies to human immunoglobulin and assayed to flow cytometry. Fluorescence microscopy. Positive fluorescence of dead cells indicate the presence of anti-donor antibodies which may lead to hyperacute rejection of graft. Donor lymphocytes, recipient serum, complement, fluorescent anti-human IG followed by flow cytometry, fluorescence microscopy. Basis of cross-matching. Then tissue typing can be conducted like antibody-mediated microcytotoxicity test. The presence or absence of various MHC alleles can be detected using this test like antibody-mediated microcytotoxicity test. In this test, white blood cells WBC from recipient and donor are taken in various wells on a microtiter plate and antibodies against various MHC alleles are added along with complement proteins. The lysis or cytotoxicity is then assessed using vital dyes such as trypan-blue. If a WBC express a particular MHC molecule for which specific antibody is added along with the complement, cell lysis takes place which is further detected by uptake of trypan-blue. Hence, HLA typing can be done by this test in order to detect histocompatibility. HLA-A allele donor, addition of antibody to HLA-A allele, addition of complement, addition of trypan blue, HLA-A allele 1, recipient, donor and donor. HLA typing of antibody mediated microcytotoxicity test. Here the donor A shares HLA-A antigens recognized by anti-sera in wells 2 and 8 with recipient, whereas donor B doesn't share any. Then mixed lymphocyte reaction or MLR. This test is used to quantify the degree of compatibility in MHC class 2 of donor and recipient. The lymphocytes from recipient serve as responder cells, whereas the lymphocytes from donor serve as co-stimulator cells. These stimulator cells are X-ray irradiated or treated with mitomycin C. T cell activated, which is indicated by proliferation of recipient T cells, is measured by intake of H thymidine in DNA. Greater the H thymidine intake means greater the cell 2 MHC difference in donor and recipient. The intense proliferation of recipient's lymphocytes indicate poor prognosis of survival of graft. Then immunosuppression, general and specific. Some allelic mismatches and histoincompatibility are always observed, which require suppression of immune system of the recipient. But the immunosuppressive drugs have many disadvantages. Like most of these drugs are generalized immunosuppressive in nature, and hence they act against all the antigens and not only antigens present in allograft only. Most of the drugs are generalized immunosuppressive in nature. Then most of these drugs are aimed to slow down the proliferation of activated lymphocytes, but they actively driving non-immune cells like hematopoietic stem cells in bone marrow, epithelial cells in gut, etc. also get affected, which causes many health complications. Cytotoxic mitotic inhibitors, azathioprine, imuran. Then cyclophosphamide, methotrexate. Oh my god. Methotrexate, which you might have already learned or heard about, is currently on news and all. It's a folic acid antagonist that block purine synthesis. Methotrexate is a folic acid antagonist that blocks purine synthesis, and that is an immunosuppressant thing. Then dexamethasone, methyl prednisolone, then prednisone, etc. These are called like immunophilins, cyclosporin A, rapamycin, X-ray radiation. Since lymphocytes are sensitive to X-rays, recipient's spleen, thymus, and lymph nodes are irradiated with. Since lymphocytes are sensitive to X-rays, recipient's spleen, thymus, and lymph nodes are irradiated with 3 rats at the rate of 200 rats per day of several weeks before transplantation to completely remove lymphocytes. The lymphoid stem cells from non-irradiated bone marrow proliferate and renew the lymphocyte pool. Considering the lack of specificity associated with the general immunosuppressive drugs mentioned above, there is a need to develop specific immune suppression to allografts only. In animal studies, it has been achieved using monoclonal antibodies as soluble ligands reactive with cell surface molecules. The monoclonal antibodies can be used to deplete the recipient of a certain broad or specified cell population. The antibodies against CD3 of TCR complex are used, which delete the circulating mature T lymphocytes. This technique is further refined by coupling diphtheria toxin to the antibody. The cell with which antibody binds internalizes the toxin, causing its death. Monoclonal antibodies against high affinity IL-2 receptors are also used as yet another depletion strategy. Since these receptors are present on activated T cells, antibodies against them are introduced after grafting. These antibodies then block the proliferation of T cells against alloantigens present on the graft. Besides these depleting strategies, non-depleting monoclonal antibodies are also used for better graft survival. An example of the same is anti-CD4 monoclonal antibodies, which do not deplete CD4 plus T cells but induce the T cell to enter in a state of immunosuppressed state. The simultaneous administration of monoclonal antibodies against adhesion molecules ICM1 and LFA1 for 6 days after transplantation has resulted in indefinite survival of cardiac graft between allogenic mice. And monoclonal antibodies against cytokines like TNF-alpha, IFN-gamma, and IL-2 have also shown better graft survival in bone marrow and cardiac transplantations. Then to block co-stimulatory signals. T helper cell activation require co-stimulatory signal in addition to signal mediated by T cell receptor. B7 and CD28, CDL4 and CD40 and CD40L, CD15-4 are examples of such a co-stimulatory signals. By blocking these co-stimulatory signals at the time of transplantation, a state of anergy is induced in T cells against graft which suppress graft rejection. In special cases of fetal immunoglobulin and concept of immunogenically privileged sites. Concept of immunologically privileged sites. There are some sites in the body at which transplanted allografts are not rejected despite of genetic mismatch between recipient and donor. These sites lack lymphatic vessels and sometimes even the blood vessels, because of which alloantigens of graft are unable to sensitize the recipient's lymphocyte and hence usually accepted. Some of such sites in humans are cornea of eye, testis, uterus, and brain. Then fetal transplantation is a special case. The fetus carries HLA alleles from both the parents, and thus in an allograft for maternal tissue, the various possible mechanisms that prevent the rejection of fetus may include complement regulatory protein produced by the cytotrophoblast inhibit the effect of antibodies directed to paternal antigens. Then indolamine 2,3 dioxygenase is produced by syncytial trophoblast, which degrades tryptophan required for T cell activation. Then cytotrophoblast produces IL-10 that possesses immunosuppressive properties. The expression of conventional HLA class 1 molecule by syncytial trophoblast is lacking, which is required for T cytotoxic cells. The cytotrophoblast express unconventional HLA-G, which acts as a ligand for KAR present on NK cells. And clinical transplantation, common organs, limitations, and possible solutions. A really quick intro. A number of factors that decide the frequency with which an organ or tissue is transplanted are availability of that organ or tissue, clinical requirement of transplantation, grade of difficulty in transplantation process, then post-transplantation caring, factors affecting acceptance or rejection of transplant. Some of the clinical transplantations in practice or in development along with common reasons are, here we could see keratoconus, keratitis, dystrophies in case of cornea, heart failure, heart, terminal renal disease in kidney, cystic fibrosis, pulmonary hypertension in lungs, cirrhosis, cancer, biliary atresia in liver, immunodeficiency, leukemia and bone marrow stem cells, burns, skin, diabetes mellitus, pancreas, Parkinson's disease, neuronal cells, intestinal failure, cancer, small bowel. Organs shown in the blue are routinely transplanted ones, whereas the organs tissues shown in yellow are still not in routine. Limitations of possible solutions of clinical transplantations. The success of clinical transplantation is limited by two major issues. Shortage of immune organs. There is always a high demand of organs or tissues for clinical transplantation, whereas the donor pool is very limited. Hence, alternative approaches need to be investigated which fulfill this demand, such as development of artificial mechanical organs, therapeutic cloning and tissue engineering to develop artificial human organs, xenotransplantation. Then chronic rejection of transplant. It results in continual damage to transplanted organ and this necessitates the immune suppression of recipient, which may lead to increased incidence of infection and malignancy. In order to resolve it, tolerance needs to be induced in the recipient for a graft. We need to develop assay to develop tolerance level and translate therapies developed in animal model to clinical practice. Now, another summary. Let's have a summary. Transplantation refers to transferring of cells, tissues, or organs from one site to another. The tissue being transferred is called as graft, and receiving individual as host or recipient are the one from whom graft is taken as donor. So, graft rejection displays all the hallmarks of the adaptive immune system reactions, such as specificity, memory, and non-self recognition. The mechanism of allograft rejection is T cell mediated. Cell mediated immune response to alloantigens present on the cells of grafted tissue organ in the cause of graft rejection. It involves a cell mediated cytotoxicity and delayed type of hypersensitive reactions. The process of graft rejection is divided into sensitization stage and effector stage. In the sensitization stage, proliferation of lymphocytes in recipient takes place against alloantigens present on the graft, whereas in effector stage, effector mechanisms take place which lead to destruction of graft tissue or organ. According to degree of variation between donor and recipient, grafts can be classified as autografts, isografts, and allograft or xenograft. The transplanted tissues which are not rejected or which do not induce an immune response are called as histocompatible. So, histocompatibility results due to antigenic differences. These transplantation antigens include blood antigens, major histocompatibility complex antigens, and minor histocompatibility antigens, non-ABO blood antigens, HY antigens. Depending upon the organ or tissue being transplanted and the immune reactions involved after rejections have different time courses, and accordingly they are classified as hyperacute rejections occurs within 24 hours of transplantation. Acute rejection begin by first few weeks after transplantation. Chronic rejections start from months to years after transplantation. Graft rejection can be avoided or minimized by adopting various mechanisms such as familiar grafting, cross matching, tissue typing, and use of various general and specific immunosuppressive drugs. Various tissue organs are in routine use in clinical transplantation practice, whereas many others are being developed. Shortage of donor organs and chronic rejections are major barriers for success in clinical transplantation. Some organs are privileged in terms of immune barriers and hence do not face immunological rejection issues like cornea, uterus, brain, testes, etc.